Hello all, I'm going to talk about the third way of gene transfer seen in bacterial cells, transduction. The definition for transduction is, it is a form of horizontal gene transfer in which a virus carries a chromosomal DNA fragment from a donor bacterial cell to a recipient bacterial cell. Simply speaking, here, using a virus, DNA will be transferred from one bacterial cell to another bacterial cell. Now, since virus is involved here, we need to know a little more about the viruses that carry out such a gene transfer. So let us see that now. The virus participating in transduction is called a bacteriophage. That means this virus should be able to enter inside the bacterial cell. It should be able to multiply inside the bacterial cell and come out. So such viruses which grow in bacterial cells are called bacteriophages. Now, just like any other viruses that you might be knowing already, the bacteriophages are made up of two important components. One is it might be having either DNA or RNA as its nucleic acid, and the other one is it will be having a protein coat. The structure and also the replication of viruses you will be studying in detail you would have studied in detail in the second semester. So please go through that to understand this part better. Now, as you may be knowing already, bacteriophages replication cycle may be of two types. One is lytic cycle, the other one is lysogenic cycle. How these cycles happen, we will be seeing in the next slide. But before that, just know these terms now. Lytic cycle is carried out by the bacteriophages called virulent phages, and the lysogenic cycle is carried out by the bacteriophages called temperate phages. Now, another term we should be knowing is prophages. What are prophages? When the phage DNA during its lysogenic cycle integrates into the bacterial chromosome, it is called a prophage. Now, this is the diagrammatic representation of the lytic cycle carried out by bacteriophages. You might have studied already in the second semester, but just to refresh your memory, I am showing this slide to you. Now, what happens during lytic cycle is the bacteriophage comes and binds on the surface of the bacterial cell. Now, after adsorption, penetration of DNA happens and the phage DNA alone will enter inside the bacterial cell and reaches the cytoplasm of the bacterial cell. Once the phage DNA enters inside, it takes complete control of the bacterial cell and the bacterial DNA will be broken down into pieces. At the same time, the phage DNA will undergo replication continuously. After that, many phage particles progeny phage particles will be formed inside the bacterial cytoplasm, and once it reaches the maximum number, finally the cytoplasm will burst open and the progeny phage particles will come out of the bacterial cell, and these progeny phage particles will now undergo another cycle of lytic cycle. So this is the basic events that happens in the lytic cycle. Now what happens in the lysogenic cycle is the first two steps will be the same as lytic cycle, where the adsorption of bacteriophage will be happening on the surface of the bacterial cell and penetration happens where the bacteriophage DNA will be entering inside the bacterial cytoplasm. Now, after the second step, here the phage DNA will integrate itself with the bacterial chromosome or bacterial DNA. So, this is the third step where the phage DNA has integrated with the bacterial chromosome. So, in this state, the viral DNA will be called prophage, and then in this stage, the viral DNA will be carried from one generation to next generation for many generations together. That is, when the bacterial replication happens, along with the bacterial DNA, the phage DNA will also get replicated. So in this condition also, the viral DNA will undergo multiplication. Now, after many generations, because of some stress or some type of trigger, the viral DNA will come out of the bacterial DNA. It could be UV light or it could be some other trigger. So at some point the viral DNA will come out of the bacterial DNA and will carry out the normal lytic cycle what you have seen before. So this is how the lysogenic cycle will happen. There are two types of transduction that is seen in nature. One is called generalized transduction and the other one is called specialized transduction. First, let us see what is generalized transduction and how does it take place. Basically, generalized transduction was accidentally discovered in 1951 by Zinder and Lederberg, and generalized transduction usually occurs during the lytic cycle of virulent phages. That means uh, uh, I already told you what are virulent phages, and also you know already what is a lytic cycle. So usually this type of gene transfer happens during the lytic cycle of virulent phages. Sometimes temperate phages also can carry out, but very commonly it is seen in the lytic cycle. And why is it called generalized transduction? The reason is any part of the bacterial genome can get transferred from one bacterial cell to another bacterial cell using this phage. So that is why it is called a generalized transduction. Now the other points what you are seeing in this slide are all the events that takes place during the generalized transduction. Just for continuity's sake, I have put these points in this slide, but you might not understand it at this stage. In the next slide, with the help of a diagram, I will explain the same events to you. So what happens during the generalized transduction is, after the phage injects the DNA into the cell, the host cell chromosome is digested into small fragments. This happens in any lytic cycle, right? So once the phage DNA enters into the bacterial cell, what it first does is it will digest or it will break the host cell DNA into small fragments. Now in the second stage, when the new phage DNA is produced, the DNA normally is packaged into the new phage particles. This also you have seen already what happens in the lytic cycle, where the viral DNA will be packaged into the viral head to give rise to new progeny phages, right? Now in the third stage, a rare event happens. What is it? On rare occasions, that is 1 in 10 to the power of 8 cells. In 10 to the power of 8 cells, one cell might undergo this rare event. What happens here is a random fragment of a bacterial DNA may accidentally be captured in the packaging process and end up in a phage head with the bacterial DNA rather than the phage DNA. As I said, you will understand this when we see the next slide, so don't worry. And these phages are fully formed and they can infect another cell, but they do not carry any phage genes, and so they cannot replicate themselves after infecting a new cell. So that is why these phages which carry the bacterial DNA will be called defective particles. So this is the continuation of the previous slide. After getting the bacterial DNA by mistake, what happens is they will be released from the host cell after lysis, and the defective phage particles will attach themselves to a new recipient cell, new recipient bacterial cell, and as usual they will inject the DNA inside this new recipient cell. But the DNA what is entering inside is not a phage DNA but a bacterial DNA. Once in the recipient, the new genes can undergo recombination with the recipient's DNA, because the DNA that has entered inside actually is the bacterial DNA, some part of the bacterial DNA. So this part might have a similar DNA or a similar fragment in the host cell chromosome also. So as usual, they will undergo recombination. So the recipient has now been transduced using the genes from the donor cell. So basically what happens here is a bacterial DNA has been transferred from one bacteria to another bacteria, but not by conjugation, not by transformation, but by using a virus. So that is why it is called transduction. transduction. This phage which has done this process is known as the generalized transducing particle or generalized transducing phage. So whatever I have said in previous two slides has been shown in a diagrammatic way here. Just observe this. The phage has come and bound on the surface of the bacterial cell. Just for understanding, let us see these two genes here. In the bacterial DNA, we have his gene and also lyse gene. The viral DNA is entering inside this bacterial cytoplasm, and once it enters inside, as I said, it will break the bacterial DNA into smaller fragments, and then it will undergo replication. Phage DNA will undergo replication continuously, and in the next step, usually the phage DNA will be entering inside the phage head and a complete phage particle will be formed. You see three phage particles here which are complete in nature, inside which you see the phage DNA. The red color DNA is the phage DNA which has entered inside the phage head. But the fourth phage particle, look at this fourth phage particle. This also is a complete phage particle with head and tail regions. But there is just one difference here. Inside this phage head, you don't see the phage DNA, but you see the bacterial DNA. The blue color DNA is the bacterial DNA, isn't it? So the phage, instead of phage DNA, the bacterial DNA has entered inside, which is basically the his plus DNA, or his DNA has entered inside this phage particle. So as I said, this is also a complete phage particle. So it will come out of this lysed bacterial cell and it can go and bind on the surface of another bacterial cell, and it can undergo penetration where the DNA will be entering inside another recipient bacterial cell, another bacterial cell. Fine. But what is entering inside in this case is not the phage DNA, but the bacterial DNA, especially the his gene has entered inside. Fine. Now, since this is a bacterial DNA here, it will have a similar DNA in the recipient cell also. Recipient is also a bacterial cell. So recipient cell will also have a similar gene like this gene. His gene is here, his gene is also here. But the only difference here is, just for our understanding, they have shown it as his minus gene, whereas here it is his plus gene. So they will undergo recombination so that in the place of his minus gene, the his plus gene will come and integrate. So the bacteria, this recipient bacteria has undergone some kind of change here because of the DNA that has been entered inside through the bacteriophage, isn't it? So what has happened basically here, the gene of the donor bacterial cell has entered inside the recipient bacterial cell and has changed the recipient bacterial chromosome. So this is what we call transduction, because here to transfer this his plus gene into this recipient cell, a bacteriophage is involved, a virus is involved, right? So that is why it is called transduction. But why do we call it generalized transduction? Because any of this fragment, provided it has the proper size, any of this fragment can enter inside the phage head. It need not have to be only his gene. Just for explanation, they have shown his gene, but any such fragment can enter inside the phage head. Since any of the fragment can enter inside, we call it as generalized transduction. And please go through the previous two slides to understand it in words. Now let us see the second type of transduction, that is specialized transduction. It occurs as a result of lysogenic cycle. As I already told you, generalized transduction happens during the lytic cycle of virulent phages, whereas the specialized transduction occurs during the lysogenic cycle of temperate phages. Okay, and why do we call it specialized transduction? Because here there will be transfer of specific genes happening. There any gene can get transferred, whereas here specific genes are transferred. And as I said, the temperate phages carry out this type of transduction, and one very commonly studied example for specialized transduction happens during the replication of this particular temperate phage, that is phage lambda, which usually infects E. coli cells. Okay, now and the other details what I have shown here are all the events that takes place during the specialized transduction. So just I will read it out. Later on we will see that in detail in the form of a diagram. Now, being a temperate phage, the lambda phage DNA will integrate as a prophage into the E. coli chromosomal DNA, and at some time in the future the prophage will undergo excision from the E. coli chromosome and it will enter the lytic cycle. These two events I have already told you when I talked about lysogenic cycle. So the same events are again told here. Now on rare occasions, when this lysogenic cycle is happening, on rare occasions an improper excision might be happening. Improper or imprecise excision might happen, excision of viral DNA from the bacterial DNA. So sometimes improper excision will happen and the excised prophage will take along a few adjacent bacterial genes while leaving behind a few phage genes. That is, it will take some bacterial genes with it and it will leave some of its own gene with a bacterial chromosome when the excision is happening. Now after excision, again the same lytic cycle events will happen. At the conclusion of phage replication, multiple copies of the phage gene will be formed along with the donor gene in it. Okay, these are called defective phages because they are missing a few phage genes needed for their own replication, because they have left behind some of their own genes with the bacterial DNA, right? So they are going to be called defective phages again. So such a transducing phage can infect another cell and transfer its genes, but they cannot undergo replication. Instead, the genes will integrate into the recipient chromosome where it will be carrying the donor's gene with it. As before, the recipient cell acquired genes from the donor bacterial cell and the recipient is now considered transduced. So this is again about specialized transduction in the diagrammatic representation. The first two events are not shown here. The first two events of lysogenic cycle, which we have already seen. The bacteriophage will come and bind on the surface of the bacterial cell and penetration of phage DNA will happen, and once the phage DNA enters inside, being a temperate phage, it will go and get itself integrated with the bacterial DNA and now it will be called prophage. And during excision, that is after triggering, during excision, the phage DNA will come out of the bacterial DNA, but this excision will not be proper. It will be an imprecise or an improper excision. So at the end of excision, what you are seeing here is the phage DNA along with the adjacent bacterial DNA, small segment of bacterial DNA which is shown in blue color will come out along with the phage DNA, but at the same time some fragment of the phage DNA will be left behind in the bacterial DNA itself. So this is a defective phage particle which has come out. Now after it comes out, the normal events that happens during lytic cycle will continue, where many phage DNA will be formed by multiplication and the bacterial DNA will be cut into pieces, and what happens in the next stage we will see in the next slide. So again, the same picture here. The phage DNA would have undergone multiplication, but when the phage DNA undergoes multiplication, a small segment of the bacterial DNA would have also undergone multiplication here. Now, in the next step, assembly will take place and also release of phage particles will happen. But here, this phage progeny phages will be carrying the phage DNA along with the small segment of the bacterial DNA, specific bacterial DNA which is present near to the phage DNA. So only that piece will be present here. So this is a defective phage particle which will be formed in large numbers. Now these progeny phage particles, when they come out, they will go and bind on the nearby recipient cells and they will inject their DNA inside. But what is entering inside is a mixture of phage DNA and the bacterial bacterial DNA. So again, the bacterial DNA will find a similar partner here in the recipient bacterial DNA. So in that case, they will undergo recombination where the bacterial DNA will integrate itself with the recipient bacterial DNA. That is, the donor bacterial piece will undergo recombination with the recipient bacterial DNA. So you will find a stable recombinant here. Fine. So again here, uh, this event can also occur where this prophage this phage DNA will undergo integration again as a prophage in the bacterial chromosome. But this event, it does not bother now. Just understand this event, that is enough. That is, the viral DNA along with the bacterial DNA will enter inside the recipient cell, and if it finds a partner or if it finds a similar fragment in the recipient bacterial DNA, then it will undergo recombination and it will be transferred to the recipient bacterial DNA. So again here also this blue color is the donor DNA, right? So the DNA has been transferred from a donor bacterial cell to a recipient bacterial cell, but not directly, with the help of a virus. So this is called again transduction. But why is it called specialized transduction? Reason is, as I said, only those particular DNA fragments that is present next to the prophage. Prophage is the viral DNA which is in integrated form with the bacterial chromosome, isn't it? So only those bacterial DNA which is present next to the prophage DNA will be transferred here, not any other fragments of DNA, and that is why it is called a specialized transduction. Now, for those who want to know more about specialized transduction, I have included this slide. For others, you may omit this slide if it is confusing. Okay. Now, what has been shown here is, this is the red color circle is the phage gene and the blue color one is the bacterial gene. Once the phage gene enters inside the bacterial cytoplasm, as I said, in lysogeny, integration of the phage gene will be happening inside the bacterial chromosome. This integration happens especially in a region called at site, present in both the phage genome and bacterial genome. The yellow color one and the blue color one, here what you see is the same site present in both phage gene and the bacterial genome. Okay, this especially is seen in lambda phage and on E. coli genome. Okay, so with the help of this AT site, the viral gene will integrate itself with the bacterial chromosome. So this is the integrated prophage that you are seeing in 1, 2, 3 configuration, and near to this prophage, what you see here in bacterial chromosome is the gal plus gene and the bio plus gene. Gal plus gene is the galactose utilization gene and this is biotin synthesis gene. So these two genes are present adjacent to the prophage gene in the bacterial chromosome. Right? Now, in a normal excision, what will happen is this whole phage gene will come out of the bacterial gene or the bacterial genome, and a normal phage gene now will undergo the lytic cycle. This happens in the normal excision process. Whereas in the improper excision process, what we have seen in specialized transduction, what you see here is an improper excision or imprecise excision happens, and when the phage gene comes out of the bacterial gene, see here, you find a small piece of the bacterial gene here present along with the phage gene. So phage genes 1, 2 segments are there, but third segment is not there. Why? Because the third segment has stayed behind with the bacterial genome. Whereas a part of the bacterial genome, this part would be either this region or it would be this region. So here in this diagram, what they have shown is the gal plus region. This particular region has come out along with the phage gene. Now this is a defective phage. defective lambda phage, because as I said, this is a lambda phage that we are talking about. So that is why this particular viral DNA is called lambda D gal. Lambda is the phage name, D is for defective, gal is for the gene that has been taken from bacteria. So lambda D gal. Now this defective phage particle again will undergo all the other processes, that is multiplication and packaging, and then the phage particle that will carry this lambda deagle again will undergo lytic cycle where the phage DNA will be entering inside a new bacterial cell, that is a recipient cell. Now this is the recipient cell, bacterial cell DNA. Now if the recipient bacterial cell is a gal minus cell, that means it cannot utilize galactose, that is gal minus cell, and when this lambda D gal enters inside this recipient cell, there is gal plus gene here and there is gal minus gene here, so they are similar in nature, so they will undergo recombination, and this gal plus gene get integrated in the place of gal minus gene. Now this is a stable transductant because here the gal plus gene has been introduced by the phage DNA in the recipient bacterial chromosome. So these are the steps that you can see again in detail, where the phage DNA carries the donor bacterial DNA into the recipient cell and changes the chromosomal nature of the recipient cell, becoming a stable transductant. So this here, since only a specific gene is carried, that it could be either gal plus gene or bio plus gene. Since only a specific bacterial DNA is carried out here, it is called specialized transduction. You may omit this part for now because you might find it a little more confusing. So just concentrate on these events. Okay, now what is the significance of this transduction? Does that happen in nature? Yes, it happens. Especially the generalized transduction happens very commonly, and some specialized transduction is a very extremely rare event in comparison to the generalized transduction. But specialized transduction has its own significance. For example, you will be studying about Corynebacterium diphtheriae, which causes a disease called diphtheria. And for the diphtheria disease, a particular toxin has been released by the bacteria, and that is called diphtheria toxin. And this diphtheria toxin from the Corynebacterium is mainly coded by the viral DNA, which is present as a prophage inside the Corynebacterium. So the information for the toxin production by the Corynebacterium is provided by the prophage DNA or proviral DNA, which is present in the integrated form in the Corynebacterium cell. Same way, there are many other toxins which are encoded by this prophage DNA. For example, the staphylococcal enterotoxin, which is responsible for food poisoning in human beings. The clostridial toxin, which is responsible for botulism, again a food poisoning agent, and also streptococcal toxins, which is responsible for scarlet fever. So all these toxins are encoded by the prophage DNA that is present in the integrated form in the bacterial cells. So we have seen three different forms of horizontal gene transfer, which is resulting in genetic recombination. Now just compare these three types of horizontal gene transfer, that is transformation, conjugation and transduction. Pause the slide and go through this slide. Just when you compare it, you might understand it much better. So what happens is in transformation is a dead bacterial cell will release its DNA in the surroundings, and these bacterial DNA will be taken up by the recipient cell and it will undergo recombination. This is one way. The second way is conjugation, where two different bacterial cells will get connected with the help of conjugating plasmid, that is the F factor, and during this in this conjugation bridge, the bacterial chromosome from the donor cell will pass on to the recipient cell, that is from a F plus cell to an F minus cell. So this is the second way of gene transfer. And the third way, where we have two types of transduction called generalized and specialized transduction. A virus will be involved. It could be a virulent phage or it could be a temperate phage, which will transfer the DNA from the donor cell to the recipient cell. So again you will get a recombined recipient cell. So just pass this slide and go through it to compare and understand the three forms of horizontal gene transfer in a better way.